This is lecture 3. In lecture 2, we went over some basic principles of exercise physiology. In this one, we'll talk about these three things: the basic function of skeletal muscle and its structure, both big stuff and tiny stuff. So you have a lot of skeletal muscles, not just the muscles in general, but skeletal muscles. And most of them you've never thought of. Some are anomalies like supernumerary muscles, what you have in excess of the nearest person to you. We talked about one of those in the previous lecture, the palmaris longus. You might be able to see its tendon in your wrist, but there are others too. The plantaris in your calf, or maybe not in your calf. It depends. There's no easy way to tell. Maybe 10 to 15% of people don't have that. Reported ranges vary and don't really matter. Or the psoas minor, that's in your pelvic region. Maybe a third of people don't have that one. Again, ranges vary. But even if you don't have those, you have tons of other muscles, face muscles and vocalization muscles. There's muscles everywhere. There are a couple hundred obvious ones. Here are a bunch of muscles in your arms and your legs. You don't need to know any of them. This is just illustrating the abundance of unique skeletal muscles. But there are also less obvious muscles, like a little bump on the very inside of your eye next to your tear duct. If you don't have a third eyelid, it's pointless. We also have auricular muscles to move around our ears, towards sounds like cats do. But our ears don't move like cats. The point is, we have a lot. We have a lot of muscles. Some of them are critical to human function. Others are pointless vestiges, useless little, lumpy relics. The purpose of your skeletal muscles, though, this is to move your bones around. We're not plants. Okay, plants can't move, with the exception of things like a Venus flytrap, but they're not moving with muscles. It's the movement of water causing a change in the volume of its lobes, and that's not how people move. Although it does have these small hairs, and those serve as mechanosensors, and we have mechanosensors too, which will be a big topic later in this semester, but I'll talk about one of them later in this lecture. For the most part, though, plants can't move. They can't go collect food. They can't run from predators. They can't swat away pests. Plants have to synthesize pesticides to protect themselves. Things like sulforaphane. When we ingest sulforaphane, we actually upregulate our body's own natural defense system. We get a chemoprotective effect. We'll talk about this later in the semester. Um, when we get to the stress physiology stuff. But what's interesting is that the protective effect of sulforaphane against carcinogens is highest if you eat it first and then experience the exposure. But the point is, we're not plants. We don't synthesize pesticides. We move. We have to move to get our food or to, fight or flee. Survival depends on movement. And if you look at a diagram, how do you tell what the muscles do? How can you tell a muscle's function? You just look at its line of pull. And then how do you tell what the line of pull is? Look at its origins and insertions, where it attaches to your bones. And then there's a joint that'll cross, and the muscle moves that joint in the direction of the line of pull. That's what your skeletal muscles do. Now, muscles come in all shapes and sizes. There are big, thick ones like your glutes that have a really high capacity for force development. Then there are long muscles, long and usually slender muscles like your hamstrings. Those have a faster velocity of shortening and a greater total amount of shortening they can do, but they're not quite as much, they're strong. There's not quite as much force production as a glute. But it's not just long and slender versus thick and bulbous. There are different shapes entirely. And the three that I'll have you pay attention to are the fusiform, parallel, and pennate. All categories of pennate, right? Unipennate, bipennate, multipennate. We'll lump those together as one. And the difference between parallel and fusiform is that parallel is thick at the top, it's thick at the bottom, it's thick in the middle. That's parallel. And fusiform is spindle shaped. That word spindle is going to make a reappearance in a future lecture, spindle. But what it means is the origin and insertion, that's narrower in those parts. And the muscle belly is much wider. The biceps brachii, you have a bicep in your hamstrings, but the biceps brachii, the one in your arms, that has a spindle shape that's fusiform. And then the pennate, this has angled fibers. The muscle fibers don't run in the same direction as the line of pull, not exactly in the same direction as the line of pull. Parallel and fusiform have muscle fibers that run exactly in the direction of that line of pull, but pennate does not. It's diagonal. These different shapes of muscles determine their function. So think about street parking. You're going to park your car on the street, right? There's parallel parking. You can't get that many cars on the road, but they're all aimed in the direction of the road so they can sort of dart off in a hurry. And then there's the pennate fibers. And this is, diagonal parking where you can get more total fibers. More cars can park along one stretch of road if they're aimed diagonally. But if we were to put it in gear and like gun it, they wouldn't move in the forward direction very fast. But there's an awful lot of engine parked on that road. So they're stronger, but a little bit slower, if that analogy makes sense. Now, if you cut open a muscle, you'll find that everything is segregated by fibrous layers, this collagen-rich connective tissue. If you go to the store and buy meat, fish or beef or whatever, it'll have saran wrap over it. And we do too. Our muscles do too. And not just our muscles, like tons of things in the body are segregated by saran wrap. But sheathing the entire muscle is the epimysium. And then inside of a muscle, inside of a whole muscle, is a bunch of fascicles. And fascicles are bundles of muscle fibers. And muscle fibers are cells. Okay. A muscle fiber is a cell. And around that cell, around that individual cell, the muscle fiber being the cell, there's a endomysium, a fibrous sheath. That's the endomysium surrounds the cell. And then around a fascicle, there's a paramysium. Um, so from the outside in, right, from the whole muscle to the fascicle, to the fiber, going from the outside in, it's the epi, peri, endo. Epimysium, paramysium, endomysium. Epi, peri, endo. This is just another look at the exact same thing. What's called a fasciculus here, that's a fascicle. Sometimes you'll see fasciculus and fasciculi very occasionally. That's Latin for little bundle. Just call them fascicles. But inside of a fascicle, we have a bunch of muscle fibers. Inside of the fibers, we have our myofibrils. We're getting into microstructural contents of a muscle fiber here, no longer in the macro structures. We're getting micro now. So again, there's the whole muscle. A whole muscle contains a bunch of fascicles. Fascicles are bundles of muscle fibers, and muscle fibers are cells. A muscle fiber is a cell. And inside of that cell, we have myofibrils. Myofibrils are longitudinal strings of sarcomeres, which contain the myofilaments, actin, and myosin. So we'll return to the topic of actin and myosin in just a minute. But first, let's talk about the muscle fibers themselves. Throughout the body, muscles have different amount of fibers. It's not like every muscle is allocated 10,000 fibers at birth. It varies. So like the tensor tympani pictured here, that has about 100 total muscle fibers in it. What that muscle does, like if you're chewing food and it's kind of loud, it would be louder were it not for the tensor tympani's dampening of those sounds. And it doesn't need any more fibers to accomplish that. That's all it needs. By comparison, there's the gastrocnemius. Gastrocnemius, like gastro, of the leg, like the stomach of the leg. It's Greek, right? Gaster is like a belly, and meme means leg. So is the stomach of the leg. A soleus, which is also one of the muscles in that area underneath, that's Latin. It means like a flat sort of sandal. So you have a stomach and a sandal in your lower leg, in your calf. And the medial stomach, the medial gastric meatus has over 1 million muscle fibers in it. And so that is a lot. And you need a lot there. That's why it has so many, is need determines their placement. Um, there are different amounts everywhere throughout the body. This isn't an anatomy class, so you don't need to know any of these muscles, but we'll touch on them for the sake of context. The first lumbrical, that's in your hand, right? Your index, the back of your index finger, pretty much right next to the first dorsal interosseous, which is between your thumb and index finger. There's about 10,000 fibers in the lumbrical and 40,000 in the interosseous. The platysma is shown here. Well, shown listed here. Um, Pull the corners of your mouth down, all like wide and hard. Just make your mouth as wide and low as you possibly can. And that weird, like neck, like straining sheet of muscle, that's your platysma. 27,000 fibers in it, give or take, but a very sort of a wide sheet of fibers. The external rectus, that's eyeball stuff. Similar fiber count to the platysma, but it's not a broad sheet that spans your whole neck. The sartorius, that's the longest muscle in the body. There's over 100,000 in that one. But the point of saying this is just that muscles are really different in their shape and the size and their fiber content. All this stuff of a muscle is really different depending on where you are in the body. And so, can you increase your fiber content through exercise, though? Yeah, almost definitely, probably. Most of the muscle mass increase when we exercise is hypertrophy, which is different. Hypertrophy, that's the growth of fibers. The fibers themselves get larger, but there are circumstances when hyperplasia is likely. It's just hard to demonstrate in people unless the people volunteer to be dissected at the end. That'd be the easiest way to show it. But we can show it in animal models, and it's been shown all the time in animal models. And we have really weird ways of making these animals exercise, but we do see hyperplasia all the time. It's cats and it's mice and whatever. We see the increase in the number of muscle fibers. We have compelling evidence from much of the animal kingdom, but human evidence is limited. But with heavy loading, we do see some fragmenting. There's still some doubt on whether it's an independently governed, innervated, right, quote, bona fide muscle fiber. But if you can find this in every zoo animal, it seems at least conceded to assume that we're so special of a species that we're not part of the same animal kingdom, bound by all the same principles of muscle physiology. But that's what hyperplasia is. It's the increase in muscle fiber content, muscle fiber number. That's hyperplasia, and almost certainly humans do it. Um, Now, inside of a muscle fiber is a bunch of myofibrils, and myofibrils are long strings of sarcomeres. Sarx, S-A-R-X, that means flesh, generally muscle. Meros, that's portion or part. So a sarcomere is the portion of muscle. And then in that top row, that spans the whole screen. That's a myofibril. It's like super blown up, but and that's like a drawing, but that's a myofibril, a string of sarcomeres connected from end to end. In that picture, there are 8 total sarcomeres in a row. And a myofibril has a lot more than 8 sarcomeres, but here are 8 of them in a row. The length of a single sarcomere, uh, you know, 2.5 micrometers, something like that. That's a give or take number. The reported measurements that I've seen are all a little bit different, but you often see around 2.2. And there are, 1,000 micrometers into millimeters, so these are really tiny. I mean, these things are really, really tiny. To put it into perspective, just the sake of comparison, human hair is probably about 100 micrometers or microns in diameter, not circumference, but diameter. That's obviously give or take a lot, 150 if your hair is like fabulous and luscious and stuff. But for the sarcomeres, the length in this study, looking at the first dorsal interosseous, it was between 2 and 2.5 micrometers for the most part. Measure enough of them, and you'll see some exceptions. In this study, the authors were looking at some different muscles, the abs, external and internal obliques, transverse abdominis, and they were measuring sarcomere length to be, the mid to low twos and some in the low threes. So there's a range. Like the point of telling you this is not that you'll memorize all the numbers, but to realize that there is variance in the sarcomere length. They're not all like identical anatomical structures. So how many sarcomeres are there in a fiber? Uh, we know what sarcomeres look like and how big they are, but how many do we expect to find? In this little graph here, this list, times 10 to the fourth, that just means add 4 zeros to those numbers. The biggest number there is a sartorius, right? 17.4. So that means 174,000 sarcomeres. The sartorius being the longest muscle in the body, right? These are really long muscle fibers. The gracilis is long too. That's a long adductor. Semitendinosus is a long hamstring muscle. So there's lots of sarcomeres in those. But the medial gastroc, which we talked about earlier, about 15,000 sarcomeres, multiplied by 2.2, that'd be 33,000 micrometers, 3.3 centimeters. That's 1.3 inches about, so it's not that long of a muscle fiber. Other muscles are way longer, like the sartorius again. The quotation below is from the article listed, and 600 millimeters is about 2 feet. So that's a long muscle. And some of those fibers run the whole length. One single cell is 2 feet long. That's a huge cell, and size in this sense matters for some areas, and we'll talk about that later in the semester. But other fibers terminate what's called intrafascicularly. That just means within the fascicle. They don't stretch the whole distance. Some terms that you'll need to know about this is sarcomerogenesis and sarcomerolysis. So genesis is the addition of sarcomeres in series. So if it used to be 15,000, now it's 15,050 or whatever. More sarcomeres are added. Sarcomerolysis is the deletion of sarcomeres in series. So if it used to be 15,000, now it's 14,900, or, again, somewhere in there. A skeletal muscle is constantly, all of your skeletal muscles are constantly calibrating their contents to be fit to the profile of its uses. If what is being used for is a lot of elongated activities, it'll remodel itself so it can better perform in lengthened states. And then sarcomerogenesis is what's going to happen to facilitate that. But if you're always exercising in highly contracted positions, it'll remodel to be able to perform optimally in those contracted positions. So sarcomerolysis is what's going to happen under those conditions. Um, and keep this concept in mind. Sarcomerogenesis and sarcomerolysis and this kind of remodeling, because we'll return to it at the end of next lecture when we talk about the length tension relationship in skeletal muscle. But it seems probable that eccentric or eccentric exercise, lengthening exercise, the negative, right, of a set, that this contributes to the metabolic signals that increase sarcomeres, while concentric loading encourages the deletion of sarcomeres. So eccentric or eccentric, that would be sarcomerogenesis, and concentric would be sarcomerolysis. But it's not quite that simple. That's just like a tiny part of the picture. There are a lot more inputs on a muscle than whether it is shortening or lengthening. But this does seem to be part of the recipe for adaptation. And remember, a muscle fiber is composed of a bunch of myofibrils, and a myofibril is a series of sarcomeres. So what's the stuff that's inside of a sarcomere? Some of it's shown in this diagram. You'll have to be able to label the parts here on a diagram like this. You'll need to label the parts. So we'll start doing that. One of the most important proteins to know is myosin. Actin and myosin are the two contractile proteins, and specifically myosin heavy chain. There are both heavy and light chains in myosin. For now, we're only talking about myosin heavy chain. And this is the head, neck, and tail in the diagram. The light chains are like little necklaces worn around the necks of the heavy chains, but the heavy chain is the head, neck, and tail. It's huge. Um, and in one myosin, molecule, you have two heavy chains. And then on the head, on each head of myosin heavy chain, there's an ATPase, and that's an enzyme that consumes ATP. The other contractile protein, or contractile filaments in sliding filament theory, is actin. And actin in this picture, it's the chain of beads that you see. In the next lecture, when we talk about sliding filament theory, that's the interaction between actin and myosin, between the actin heads and these little myosin beads. But wrapped around actin is tropomyosin. That's a helical, like a helix protein that at rest will prevent myosin from binding to the actin. And we'll talk about that in the next lecture in detail in the next lecture. But along the tropomyosin, we find troponin. And this is a protein complex. There are three proteins in troponin. There's troponin T, and this binds to tropomyosin. That's what the T stands for. There's troponin I, and this binds to actin, but the I stands for inhibition. It should be called like troponin A, but because it binds to actin, but the I stands for inhibition. And so this protein it's holding tropomyosin in place along the actin. And as long as it's held there, myosin is prevented from binding to actin. So that's why it's troponin I. It inhibits the binding of myosin to actin. And then there's troponin C. That's the third one. This binds to troponin I, but it's also what calcium binds to. That's what you need to know about it. What you need to know about troponin C, C is for calcium. This is what calcium binds to. And when calcium binds to troponin C, it causes a conformational change. That just means a shape change. And that makes troponin I dislocate and the tropomyosin moves. And we'll talk about that in the next lecture. In this diagram, we see myosin in the middle. It's labeled, quote, thick filament, because that's what myosin is. It's the thick filament in sliding filament theory. And we see thin filament. That's the actin. The thin filament in sliding filament theory is actin. And then we see a line or a disc. That's the Z disc or Z line. Those are the outside boundaries of a sarcomere. And we see one more protein. It's labeled an important protein here, and that's titin. It's that squiggly one. And titin goes from the Z disc, the outside border of the sarcomere, to the M line, which is right in the middle. So it covers half the distance of a sarcomere. It's more than a micron in length, a micrometer in length. And that probably sounds small, but it's the largest protein ever discovered, not just in the body, but just like ever discovered. And it's the third most abundant in your sarcomeres behind myosin and actin. Um, several roles, right? As a bunch of roles. And one of those roles is to stabilize myosin. It also prevents the sarcomere from being stretched too far. So, like if you stretch a muscle, it gets tight, right? It gets tight. And in the way that a rubber band will, if you stretch a rubber band, that gets tight too. And that passive tension in a skeletal muscle, as you stretch it, that passive tension, it comes from the mesonems a tiny bit, right? Epiperi-endomysiums. And there's some from like, actin and myosin, but most of that passive stiffness comes from titin. But the most interesting role that titin serves is as a signaling protein. So earlier in the lecture, I talked about the hairs in a venous flytrap serving signaling functions. Titin does that too. Not to like trap flies, but it's involved in hypertrophy and muscle remodeling. It sends signals for muscle remodeling. And then this is Titan. This is the chemical name for Titan. It's less than half of it, though. Less than half of the chemical name for Titan is on the screen. It's much longer than you can see here. It's the longest word in the English language. It's almost 190,000 letters long, this word. Um, and it's kind of cheating, though, to call the word because it's just the series of amino acids. That's what you're pronouncing. And there are over 34,000 amino acids in the series. This is the chemical formula, but the pronunciation of the amino acids themselves, methionine, threonine, stuff like that, and 34,000 more, would probably take a few hours, at least a couple of hours. We can synthesize it faster than that, like translation. That's protein synthesis, translation, linking those amino acids together. That takes at least an hour, but you can probably do it faster than you pronounce it. Moving on from Titan, we have Nebulin. Nebulin is the ruler of actin, like a yardstick ruler. And it's critical to actin assembly. It stabilizes and it regulates actin's length as you're making it, as you're making that protein. So if you're missing Nebulin, you get really crappy actin. And you can see this in knockout mice. They have this short, crappy actin that's just dysfunctional. The whole muscle is dysfunctional. Uh, this is another look at the same proteins, Nebulin and many of the others that we've been talking about. You can see the Z disc and the M line. You can see actin and myosin, and you can see Titin and Nebulin there. The M line or the M band, right, as it's written here, this isn't just a hypothetical midpoint, like a center of gravity. There are other proteins that live in this area. Nothing you need to know about. The point of telling you is just to say that it's not an imaginary middle. It's a structural center made up of specific proteins. The M line has very specific proteins. Things like myomecin, and this has binding roles, adhering stuff to stuff. You don't need to know it. I'm just giving the M line its due as a real structure within the sarcomere. One last protein we'll talk about in this lecture, more to come in the future. But for this lecture, one last one, that's alpha-actinin. It's not alpha-actin, it's alpha-actinin. And these yellow, like difficult to see bits in the Z line, or the Z disc, that's the alpha-actinin. And among other things, alpha-actinin connects actin to the Z disc, so that when myosin pulls on the actin, it pulls the Z discs closer together. Alpha-actinin is a necessary protein in the structure of a sarcomere. It's connecting the actin to the Z discs. But there are four types of it. There are four types of alpha-actinin. In skeletal muscle, though, there are only two. It's just actinin 2 and actinin 3. Ignore the other two. In skeletal muscle, we only have 2 and 3, those types of alpha-actinin. Actinin 2 is in type 1 and type 2 fibers, type 1 and 2 muscle fibers. Alpha-actinin 3 is only in type 2 fibers, and type 2 fibers are the bigger, faster, stronger ones. And we'll talk about muscle fiber types soon in a future lecture. But there are a few different versions of actinin 3 that show up in a sarcomere. There's a strong form of it, there's an intermediate form, and then what's called a nonsense SNP, SNP, single nucleotide polymorphism, a SNP. But just think of it of this nonsense version as a truncated bad form of alpha-actinin. And if you're going to try to pull on something, it matters how tightly it's secured, how tightly it's adhered. So, your traction. So if you think of cross-bridge cycling, which is in the next lecture, as like myosin pulling on actin, right? If you think of that as a tug of war, doing it with the strong form of alpha-actinin is like tugging with cleats. Okay, you have an advantage. You have traction. The intermediate form would be like tug of war with sneakers, and then the nonsense snip or the nonsense form, that's like a tug of war with roller skates. You just don't have a strong connection, a strong grip. And in animal models, where you can look at actinin-3 knockout conditions, this is just selectively breeding mice to achieve this absence of the thing. The knockout mice are scrawnier than their wild type peers than the non-knockout mice. And then in people, we see similar effects, not with selective breeding, but just genetically, there's diversity in people's genes. And looking at that diversity, those with the version of the gene that encodes the stronger protein, we see stronger athletes. Uh, and we see faster sprinters. So when Usain Bolt and his fellow Jamaicans were dominating in the Olympics, geneticists were looking for an explanation. Everyone always does this. And this was one thing they came up with. It's just one thing of 1,000 that contributes to variance in speed. But this one thing does make a difference. It does make a small difference. And that's it. That's it for this lecture. So these are all of the terms that you should know from this lecture. And so words that you should be able to produce a definition for. And I will see you in the next lecture, in lecture 4, where we'll talk about sliding filament theory.